Welcome to this video on the CORMS framework. This framework is a simple way to remember all the necessary steps for designing a scientific experiment. C stands for change, the independent variable. Clearly state what you are changing and identify the specific factor you are investigating. Also, give a range by suggesting at least 5 different values. O stands for organism, a control variable. This is the biological material you are using, which must remain the same for a fair test. Be detailed by mentioning the species, age, or size. R is for repeat, which ensures reliability. You must repeat the experiment at least 3 times for each value of your independent variable. This allows you to calculate a mean and identify anomalous results. M stands for measure, the dependent variable, and typically has 2 parts. M1 is what you are measuring, such as mass or volume of gas. M2 is the time frame over which you are measuring. S is for same, the standardized or control variables. List at least 2 things you will keep constant to ensure a fair test. For example, if you change temperature, keep pH and concentration the same. Let's look at an example applying the CORMS framework, investigating the effect of temperature on enzyme activity. Here is how CORMS applies to this experiment. C is changing the temperature. O is using the same amylase enzyme and starch concentration. R is repeating 3 times. M1 is measuring time. M2 is checking every 30 seconds, and S is keeping pH constant. Next, we will go through a Model 6-mark answer, detailing the steps of the experiment. For change C, prepare at least 5 water baths at different temperatures, using a thermostatically controlled bath. For organism/material O, use the same volume and concentration of amylase and starch solution for every test. Step 3. Place 1 drop of iodine solution into each hole of a spotting tile. Step 4. Label test tubes for starch and amylase for each temperature, and place them in the water bath for 5 minutes to equilibrate. After mixing, start a stopwatch and check the mixture's color with iodine every 30 seconds. Measurement 2, M2, is recording the time it takes for the iodine to stop turning blue-black, indicating the starch is digested. For same/control S, keep the pH constant using a pH 7 buffer solution. For repeat R, repeat the entire procedure 3 times to identify anomalies and calculate a mean time. Remember to wear safety goggles and use a test tube holder. To wrap everything up, here is a cheat sheet of tips and tricks specifically designed to help you secure the maximum marks. The numbers rule suggests never leaving a variable naked. Always specify a number and a unit. For instance, instead of use different temperatures, write, quote, use 5 temperatures, 20 degrees Celsius, 30 degrees Celsius, 40 degrees Celsius, 50 degrees Celsius, and 60 degrees Celsius. Use the 30-second trick for any rate-based experiment. Checking things at regular intervals, specifically every 30 seconds, is ideal for monitoring enzyme reactions, photosynthesis, or temperature drop. For enzyme questions, use the buffer trick. State that you will keep the pH constant by using a pH 7 buffer solution. This is an easy mark for a control variable. The equilibration mark is a crucial, secret mark. Always mention letting your solutions sit in the water bath for 5 minutes before mixing them. This ensures the reaction starts at the correct temperature. Always explain why you repeat the experiment. Use the formula. Repeat the experiment 3 times to identify anomalies and calculate a mean. Mean is preferred over average. The safety freebie provides an almost guaranteed mark. Mention wearing goggles for enzymes or iodine, keeping water away from electric lamps for photosynthesis, or using a test tube holder for hot temperatures. Finally, to structure your exam answer, use bullet points or numbered steps instead of a long paragraph. This makes it easier for the examiner to mark and ensures you have covered every letter of the CORMS framework. In case you've been struggling with planning questions and this video was helpful, please subscribe, like, and share. And if you have a question you would like me to make a video on, comment the question in the comment section, and I sure shall make the video you have requested. Thank you for your continued support.